In the continuation of the previous module about the sterilization kinetics, we have one Arrhenius equation. So if we combine that Arrhenius equation with the equation which we achieved by the integration of that equation, so you can see here, so we have in that module this equation ln of nt over n naught is equal to minus kt, and k is equal to A is Arrhenius constant, E powered by E over RT. So if we combine these two equations, then we have this equation. So if we put just the value of k, this value, then we have this kind of the equation. So you can easily see that A dot E, and then this is the. So according to the D. Indorfer and Humphrey 1959, they use the term for ln naught over nt as design the criteria for the sterilization. So that factor sometimes they called as the Dell factor or Nabla factor or sterilization criteria. So represented this Dell factor or a Nabla factors are a sterilization criteria by this sign. So this sign is commonly known as Dell factor, Nabla factor or sterilization criteria. So we will use in later slides as a Dell factor. So the Dell factor is the measure of the fraction reduction in the viable organism counted, we can say that count produced by the certain heat and the time regime. So then the Dell factor, if we say that Dell factor is equal to this one, then our Dell factor is equal to ln is into n naught over nt. So this is basically show the fraction reduction in the viable organism with respect to the heat and the time of heat treatment. So as we have this equation L, and then we have this kt is equal to A dot T over this, then the delta factor is this one. So on rearranging the above equations, then the equation will be ln T is equal to E over RT plus ln Dell factor over A, that is known as Arrhenius constant. So the plot of the natural logarithm of the time required to achieve a certain Dell factor value against the reciprocal of the absolute temperature that will yield a straight line, so the slope of which is dependent on the activation energy. You can see this whole relationship in this slide. So when we draw the graph between the log of nature of the time with the temperature, which we called as absolute temperature, so then we can see from this figure, so the degree of sterilization, which we called as the Dell factor, obtained over a wide range of the time. So if we have a different time pattern, then we can say we have a 2.3, 4.6, 23 and 69.6. These are the different Dell factors with respect to different temperature. So this graph actually shows the effect of sterilization and the temperature on Dell factor achieved during the process. The kinetic description of the bacterial death enables the design of the procedures that given to the certain Dell factors. So by this way, by choosing the value of nt, then the procedure may be designed having a certain probability of achieving the sterility based upon the degree of risk. So that whole degree of sterilization and the degree of risk that totally depend upon the Dell factors. So in later module we will discuss this del factor in detail.